Okay, well, hi everyone. I'm Katie Clapp, president and co-founder of FRAXA, which you already know. And you also already know Dr. Elizabeth Berry-Kravis, most likely, who is professor at Rush University Medical Center, a site principal investigator for this trial, an advisor to Harmony Biosciences and Zynerba before Harmony, and a FRAXA scientific advisor. So Liz wears many hats, does not sleep a lot, and was really gracious to give us this time to answer questions, even though, it's super early days and these results have just come out. So we're really thankful that Liz is willing to do that. We also wanted to thank Anna DeSanya from the National Fragile X Foundation for joining us and for sharing questions that they have received from parents about the trial. So we hope to get through all the questions tonight and entertain any that you might have as we go along.
All right.
Oh boy, says Caleb.
Yeah, so I think we had a lot coming in, and the biggest thing is possibly explain like the drivers behind like the placebo effect that was seen in the study.
So I think it sounds like, I mean, I have to say up front to everyone that I haven't actually seen the data per se. I've talked with Zynerba on several phone calls, or with Harmony on several phone calls, and they did an investigators meeting. And I think they're still trying to really hash through all the data and figure out what went on. But it sounds like the placebo effect on the social avoidance scale, which was what they had elected to look at as a sign of the drug effectiveness, was very high. And it was higher than in the previous CONNECT study. And so it was sufficiently high that you would have had a huge drug effect to get over the placebo effect. And we already anticipated a placebo effect, because I think everyone needs to understand that in every Fragile X trial we've ever run, there's been a placebo effect on parent report measures as well as clinician measures, because when we're doing our interviews of, to try to find out how well the patient's doing, we're looking at the patient, but we're also interviewing the family. So placebo effects have been larger in some trials than in others, but pretty substantial. And so Zynerba had done this mitigation strategy where they, had put in they had put in a placebo period at the beginning of the study, and so that was supposed to reduce the placebo effect, but it didn't really do that. So they had put in several strategies to try to decrease placebo effect, but those strategies apparently weren't enough. And historically in trials, even when you try to reduce placebo effect, you don't eliminate it. And so I think it's really unfortunate that this placebo effect was so big. I mean, we've thought hard about why that would be, why it would be so much higher in RECONNECT than in CONNECT, their first study. And the only thing we can think of is that the CONNECT study was published, so people already, and had been presented at many meetings, so people already knew that there had been some benefit to the drug, particularly in, quote, the fully methylated group. And so going into it, if you have more of a mindset that the drug is going to help, then you may have a bigger placebo effect. And also, I was just talking about this with Katie. Sometimes placebo effect is not entirely placebo effect. It's actually a real change in the patient. Like the patient is actually better. And the reason they're better might be because they're coming to frequent visits and people are paying more attention to them. And they're just getting more sort of medical intervention and attention, or when they come to the site, suggestions are made about, things that you could do about this or that problem. And so they actually do improve. They are actually better, even though they're better not necessarily because of the drug, but because of other attention and behavioral reasons. So this is what we think caused the problem with meeting the primary endpoint. And we'll have more information as time goes on. They're still analyzing some of the other kinds of tests that the study was running. And they're very interested in looking at their long-term treatment data, because placebo effect tends to wear off, over time after the patient, isn't coming in all the time anymore, and isn't, and now the family's kind of thinking, well, I don't know if this is really working or not. And when things go on for a long time, like patients in the CONNECT study, some of them have been on this drug now for like 5 years. And so as 5 years goes on, you may kind of forget what the patient was like at the beginning. And then because you kind of have a new baseline, and then people may think, well, I don't know if this is working or not. But in this case, there were a lot of families that really felt that benefit was maintained for a really long time. So that is what Harmony/Zynerba are trying to look at now, is the whole trial, the whole 2 trials, and how they fit together, and whether there's anything there that they could, that they could make something of.
So that actually ties right into the next question I have is, so what did scientists learn from this trial, even if it didn't succeed?
Well, I think we've had a lot of trials that haven't succeeded in Fragile X, and we've learned different things from different ones. In some cases, we've learned not to do certain things, or to do certain things, or how to design a trial better. In other cases, we've learned that a drug mechanism just doesn't translate very well to Fragile X, from like mouse to man. In this case, it was CBD, so they weren't too much going off the mouse. There's some mechanisms in the mouse that suggest that CBD should be effective, but it was mostly just thought that CBD would help with anxiety, and Fragile X is a disorder with a lot of anxiety. So I think one thing we learned has to do with the FDA, and we've learned things over and over again from interacting with the FDA, but I'm not sure how much progress we've made. So when Zynerba wanted to study anxiety originally, they went to the FDA, and the FDA wouldn't let them use the anxiety measure. They had to use the aberrant behavior checklist, which, as anyone who has a person with Fragile X with a lot of anxiety knows, only taps into a small percentage of the kind of things that a Fragile X person does because of anxiety. So we're only measuring part of the problem, not the whole problem. And then that makes it a little harder to see the drug effect, because we're only measuring the drug effect in a very specific area and not across the whole range of what the problem is creating in different patients. And I think I think one of the things that we've learned kind of over and over again is that we need to come up with measures that are going to be more Fragile X specific and are not, and we're going to need to be able to talk the FDA into using some of those things, as opposed to just going back to the old measures all the time, and then having a measure that is not optimally addressing what we want to treat. And this has been an ongoing problem. I mean, has been an ongoing problem since the Seaside studies. It's like a rerun of a show that keeps going on and on with companies and the FDA and these outcome measures. And so I think we need our field needs to figure out how to break through that. And this is not just true of Fragile X, it's true of all the neurodevelopmental disabilities. We need to really have new ways of measuring outcomes. So I think certainly the one small area of social avoidance that we were measuring does not describe the whole spectrum of anxiety in Fragile X. And so what they were trying to measure, there was a lot of placebo effect, yes, but it also wasn't everything the drug might work on.
Excellent. So the next one's kind of tying into like the scientific details. So I'm not sure if you'll be able to answer it or not, but were there certain groups, age ranges, children, or like young adults that showed more benefits than others?
Yeah, I mean, we would, of course, love to know that information. And I'm sure that Harmony will be dissecting things by mutation, whether patients are fully methylated or not. And well, that was the primary outcome. They were looking at the patients that were not fully methylated. But I'm sure they'll be looking at effects in that regard. They'll be looking at age. They'll be looking at, cognitive ability level, maybe. I mean, there's different things that they can slice and dice the data by, but they haven't actually done any of that yet because they've really only had the results for like 3 days. So they will systematically sort of chop through all of that, and we'll find out that kind of information later, because they're of course going to really look for whether there's a population that they actually had a benefit for, or that they can demonstrate a benefit for. It's not that the drug necessarily didn't work. It's just that the drug effect was not possible to see based on the placebo effect. So the drug might not have worked at all, but it might have worked and just been obscured by the placebo effect.
Next question is, could you explain the methylation assay that was used to select participants?
So the participants were not selected based on methylation. Based on methylation, they allowed anyone with fragile. It didn't depend on your genetics whether you got into the study or not. So they had group, but their primary analysis was based on those that were fully methylated, and they used the Asuragen methylation PCR assay to determine that. And one could argue that people can be a little unmethylated and you can't detect it on that assay for sure. We also use Southern blot to try to figure that out, obviously. But, when we look at testing labs, we know that with the Southern blot, whether you can see a little bit of unmethylation or not depends on how hot how much you overexpose the blot so that you can see as much of the signal as possible. And then and then more recently, we have ways of looking at all the methylation sites in the gene, not just the 1 or 2 that are used in diagnostic testing. So that would certainly provide a more accurate methylation assessment, but the original study was based on a combination. The original study was based on Southern blot and methylation PCR in Flora Tassone's lab. In the RECONNECT study, we suggested they just keep the same lab, but they wanted to go to Asuragen. So that's how. But these assays should be relatively close. The only thing where you could get a really different result would be if you use the long read PCR to look at the long read sequencing to look at all the methylation sites in the FMR1 gene. And that's something that in studies, we're starting to do, but nobody's going to have that, no one would have that information, from their diagnostic testing.
Okay, next question would be, how do these results fit with other Fragile X research?
Well, if one looks at previous trials, they come out pretty similarly. The Seaside trial failed to hit its primary outcome also because of placebo effect on the same measure that was being used in the Zynerba trial. Seaside did have several other secondary measures that were significantly improved in the pediatric trial, suggesting that our baclofen does actually have an effect, at least for a subgroup of patients. However, they were never able to go and argue that with the FDA because they went out of business. And so now we've brought that data back to the FDA, and the FDA has said, oh, now it's 10 years later, you have to run a new trial. So I would say it's actually the result we're seeing here is reasonably similar to the Seaside result, where they had some things that looked good, but their main outcome didn't look good because of the high placebo effect. And in the Seaside study, we had the sense that, in fact, we knew that people were finding out what the main goal of measuring was, and Seaside was only enrolling people if they had a high enough score on the subtest and the aberrant behavior checklist they were using. And we know that some people may have been pushing up their scores a little to get in the study. And then when that happens, then they kind of revert to where they actually are over the course of the study. And the people that are reverting to where they actually are might be on placebo. And so then that contributes quite a bit to the placebo effect. So it's important to be really honest when you're rating at the beginning of a study, even if it might keep you out of a study, because in the long run, it's going to delay things more if we have people, kind of, I won't say artificially, but like making more of their symptoms than they really are, because they subconsciously know that that's the way to get in the study, and then having things kind of revert to baseline across the study. So that's something that we need to really figure out in the Fragile X field, because I think sometimes that goes on. In the Zynerba Harmony study, they were very careful not to tell people what they were basing enrollment on, how you qualified. But I suspect that there were people that got an idea of what they might have been looking for. So I don't know that. I mean, that would be something that we'd have to ask our patients.
What do you think this means for the timeline of getting treatments approved in Fragile X research in general?
Well, I think for this treatment, it obviously delays the timeline or will prevent them from being able to get, this version of CBD across the finish line. There are some CBD trials in autism, and certainly if a CBD drug got approved for autism, we would be able to use it in Fragile X, much like we do other drugs that are approved for autism. So I think there could be options for Fragile X patients to get an approved CBD if the autism studies work out, which we don't know yet. I think that this particular trial, having really started, it was before the pandemic that this started. So the CONNECT study ran like in 2018, 2019. So this has been going on for a very long time already. And I think since the time this study started, we've moved forward a little bit with our outcome measures and tried to push the FDA into letting us use things that are a little bit better for Fragile X. The Tetra study is definitely using measures that are better focused on Fragile X and the things that are happening in the Fragile X patients than the Zynerba study was. But I know when they went to the FDA, again, they wanted to have certain things and the FDA wouldn't let them do that. When Tetra went to the FDA wanted to have certain things like the aberrant behavior checklist again. And Tetra just said, no, that's ridiculous. That's not what our drug does. And so they were really the first company I've actually seen just kind of say no to the FDA's plan and say, no, we're going to go forward with what we want. And we want to work with you to validate this kind of thing, because we need to measure what's important in Fragile X and not just what's on the questionnaire. So I think we're a little more hopeful about some of the measures that Tetra has. And I think this is just another study that can be used to show the FDA we're not measuring the right things and we need to do better. And we have information that would allow us to do better, but you have to work with us on this.
So several families said that they felt that their child benefited from the study drug. So what happens next? Is there any way that they can continue getting the ZYN 000 or 002? What happens with the open label extension?
Yeah.
Are there alternatives?
So for right now, we don't know what exactly Zynerba is going to do. They're not going to do anything right away. So the open label extension is staying as it is for now. They've told the investigators that. And they don't plan to pull out immediately. They want to look through what kind of strategy they might be able to have with all the data they have over all this years. And then they want to talk to the FDA, I think. So nothing will change right now, and people can stay on the CBD. Obviously, if they go back to the FDA and talk, if there's no path forward after they get done with some of that, then it's going to be unlikely that they'll keep people on the drug long term. And there are people who benefited. I mean, we've had patients who've benefited. In fact, we've had people who were on it for multiple years and were kind of like, well, he's not as bad as he was at baseline, but maybe that's because he got older and matured, and they wanted to go in the Tetra trial. And they couldn't be in the Tetra trial if they were in the open label extension. So they came out of the Zynerba Harmony trial, and then a couple of them couldn't get in the Tetra trial, and then have had significant deterioration in behavior. We have 2 of patients like that who elected to come out of the study, thinking, well, we don't know how much this is really helping because they've been on it so long. And then, are really, that's the only change that happened. And they're really they have much more problematic behavior, probably, kind of mediated by anxiety and so forth. So I think there are some patients that are really deriving good benefit from it. But again, it's hard to know. And that's another thing that makes the trial hard to show its outcome, is that probably not. We've also had people come out of the open label extension and they've had no deterioration, or some who've even had behavioral side effects of the drug that made it kind of worse when they were on it. So again, there's some responders, there's some pretty good responders, and then there are other people that don't respond as much. And we see this with every single drug. But then because we have no way of predicting who the responders are, we enroll everybody in the trial. And so if they're 40%, if they're 30 or 40% of people with Fragile X who respond very nicely from a behavioral standpoint to this drug, that would be great to have, because we could take 30 or 40% of our patients that aren't doing all that well on what they're on, and we could expect them to have a response to this drug, which would be a big help for everyone. However, when we're doing a trial, because we have to group all the patients together, that dilutes your ability to see the benefit. So then if you throw the placebo effect onto that, and all of your benefit is based on 40% of the patients and not 100% of them, it becomes much harder to see the drug effect, because the drug effect is helping individuals, but not necessarily the whole group. So we do know there are responders. We hope it'll continue. If people are if people have to come out of the trial, we would try to find them a reputable source of CBD that they could substitute. But I'm not going to lie, getting to the doses that people are on in this study is a fairly expensive proposition from, CBD that's sold over the websites, but could be, equally effective. We just have to pick one that we're fairly confident is actually saying that. A lot of these places that sell CBD on the internet, when you assay what they say they have in their product, it's not accurate. Like, they may have anything in there because they're not regulated by the FDA at all. But we would try to pick more reputable sources of it, where they actually do voluntary testing to show how much CBD is in the product.
The next couple of questions are kind of tied to the company's roles and maybe the next steps. I'm not sure how much insight you'll really have on these, but do you have any idea of what Harmony's commitment is to Fragile X moving forward? Are they going to continue research, plan on another trial, or stopping work in this area? Again, I don't know how much insight you'll have.
I think Harmony has a strong commitment to Fragile X. I think they're very, very supportive, and they were broken about this trial. And I don't think it's just about the money. I think they really honestly wanted to be helpful. They acquired Zynerba to be helpful, I think. They thought they would, have a path forward, and Zynerba didn't have the funding to continue. So I honestly think they would like to help. I think it hurts them to think that they might have to take people off of the drug. And that's why, some companies would just say, okay, primary outcome's not positive, all the studies are ending, kaboom, everybody can be off drug within a month. We had that experience in the Fragile X field as well. But they've said, no, we're committed. We're going to keep this going for a while. We're going to try to search down all the possible avenues of maybe continuing to have a development program. And so I think there's a strong commitment there, but sometimes there's just no path at FDA. So in that case, even if you have a strong commitment, it's hard to, like, you kind of can't go on. So I think they're committed, and we'll see what they can come up with over the next months. There is one option that we've actually talked about, and we talked about this at the end of the Seaside trial as well, which is something called a randomized withdrawal trial, where you take the people who are in the open label and you take the ones who've had the best, like the ones who have the most from all the ratings, the ones who have the most effect from the drug. You have to be over a certain bar. And then you withdraw them from the drug in a randomized fashion. Like some people would go on a placebo cream and some people would stay on the Zynerba cream or the CBD cream. And that's called a randomized withdrawal. And then the way you get the drug approved is by showing that the people who had to come off the drug had worsening more so than the people who didn't actually come off the drug. So it's kind of the reverse of the way we do a usual trial. FDA used to like that design in the past, but hasn't been too fond of it in recent years. Whether they'd allow that, it's hard to know. That's always a design that's very hard on families. Nobody likes that design, that we're going to randomize you, and some of you are going to get to experience what it's like to be off the drug, and some of you are not. On the other hand, if the drug doesn't really work, then usually we won't, we might not see that much difference, in which case that would help us answer the question whether the placebo effect really obscured a drug effect or not. So, I mean, these are just ideas that are being tossed about. This is not something that anyone has decided to do. Believe me, it was tossed about for quite some time with this, in the weeks after Seaside had their initial phase 3 results, it was discussed extensively. But and it's always something that comes up when you have a trial where your data shows that there's a benefit to the drug, but your placebo group also has a benefit. So this is just one way of sorting that out. But I don't know that they will decide to do this or that it will happen. And if it does happen, I would just, I feel for the families in this kind of situation where we're doing a randomized withdrawal, because it's not a nice study, but I would just hope that people support it in order, for the long-term goal, to really find out what the effect of this drug is and see if there's an alternative path for getting it approved.
You had mentioned the Tetra Shionogi trial. There's a question related to that, was what are you seeing in that trial to the extent you can talk about, and what are your expectations from that trial drug?
Well, I can talk about the phase 2 trial because the results of that are published. And, I can say that once again, as in all trials, as in many trials in Fragile X, not everything, but many, we see some patients who seems to be doing really well in that trial, and other patients who aren't necessarily that much changed. And again, it's the same thing with all drugs, where you get people who are really good responders and people who are not so much responders. And so we'll have to sort that out with the Tetra data when they have data available. I think we're hopeful for something good from that trial. The adult trial is locked, but the adolescent trial is ongoing. So we're awaiting that data. And they'll make announcements. The database isn't locked for the adolescent trial yet, because they were still, there were still patients who had their final visit in September. So this takes a little while to clean out the data and make sure it's right. And so I think we just have to wait to find out the result. I mean, it was a very positive phase 2 trial. We've never seen a phase 2 trial where we had parent ratings of function improving, the NIH Toolbox, which is a performance-based cognitive measure, improving, and the EEG showing improvements in EEG abnormalities that are related to processing problems that we see in people with Fragile X. So we've never had a study that had all three of those things go on in the same study. So that made it a very promising medication to take forward. But again, the phase 2 study was only a small number of patients, and you have got to be able to show that this replicates in a large number of patients in order to get a drug approval. So we're hopeful. We're cautiously optimistic, but I every time when these companies come to me in the middle of a trial and they want to start working on their marketing strategies, I'm like, no, don't jinx it, because we've had enough problems in Fragile X that I kind of want to see that it's positive and that we have a hope of going forward before we start talking about marketing. So anyway, that we'll know about Tetra as time goes on. We really don't know anything yet.
Okay, so even though ZYN002 did not meet its primary endpoint, what should families take away as the hopeful progress from this study?
I think we need more of the data to be able to say that. I mean, we only know it didn't meet the primary endpoint. We don't know. We don't have other information. Like I said before, I think one hopeful thing is that eventually, if we have enough failed trials, we can talk the FDA out of the outcome measures that don't really work that well. But it may speak to additional ways of trying to mitigate placebo effect. I mean, I think right now, from what we know, the only thing we know is that the placebo effect got in the way. And so what this is telling us is that we have to find better ways of getting around the placebo effect in Fragile X.
Many parents are feeling sadness and discouragement after the news. So knowing our children already face so many setbacks, what advice do you have for families, both for coping with the emotional impact right now and for staying engaged in research and clinical trials moving forward?
I always, we had this experience before with some of the failed trials. I mean, the Seaside trial actually did show a benefit. It just didn't show the right benefit. And it wasn't, there was kind of not, the company was not able to take it forward at the time that the results needed to be argued at FDA. So I think what's happening, I mean, we all feel bad too. I've been through quite a few failed trials that I had great hope for. I've taken, out of each of those, I've taken a little something that is, okay, we should never do that again. Or this really worked and we should do this and we should develop this measure because this is a better measure. And so I think in general, the trial contributes to general scientific progress. And I think participation in other studies like the Forward study and the Toolbox study, and all these studies we have going on to try to get to that place where we have those better outcome measures are really important for people to participate in. Because even though we haven't seen fantastic movement from the FDA yet, I think we just need to keep hammering, and eventually we will be able to, have these better outcomes. So don't get discouraged about participating in research, for sure. We don't want people to get discouraged in that, because ultimately we want to be successful. And another thing is all this trial science we're learning, like how to do the trial and how to collect the measures, is all adding up to the base of knowledge that we have, that someday we're going to have actual drugs that target the genetics of Fragile X, which we haven't had yet. We have drugs that target mechanisms in the brain, and those are important because we may need to use combinations of things. But we haven't had drugs that are like gene therapy or like reactivation of the gene or anything like that. And the more we can know before we go into those really key genetic trials, the more we can know about what to do and what not to do, the better off we are. So, and the better chance we have of being able to demonstrate that those treatments work most efficiently, so that we can get them to patients as fast as possible. I mean, I always compare, when you're in basic science and you're working in the lab and you're developing like a new assay that's totally new that you don't like have an instruction book or a kit for or anything, you may have to repeat it a lot of times before you get it right. But that's, on a time scale of weeks or maybe months or something. Whereas, unfortunately, for us to get it right in clinical trials, it can be it dashes many people's hopes, and it's on the order of many years. But I think we need to persevere. We need to feel confident that we're getting better and better things that are better and better targeting the brain. And people don't see that right now because they're not meeting with all these companies that I'm meeting with that have something that's really cool and looks like it might be able to fix, at least part of Fragile X. So I think we do have good things in the future. And the more we learn now from whatever we get, the better off we'll be for our designs in the future. So don't get discouraged about research. I know we've had a long kind of traumatic route in Fragile X, but I am confident we will ultimately be successful, because I have told people I can't retire until we get a drug approved for Fragile X. So all this means is that you've got more years of me.
And we appreciate all the work you definitely do. I'm going to take a little second, just kind of do a plug, since we did bring up like family support and everything. We are hosting a Fragile X Families conversation later, middle of next month, so October 16th at 12 noon Eastern. I'll throw a link inside of the chat right now, so you guys can feel free, some of the family members, if you want to come talk to us. Myself, Holly, and Katie will all be part, and we actually all three have, over the time, unfortunately experienced a failed trial that our children were in. So we kind of have lived it. So feel free to sign up, and let's move on to the next question, which actually is a really good one. It says you were just talking about some of the other things that are in the pipeline. So what is beyond the Harmony drug or the Tetra drug, Shinogi drug? What makes you most excited about the potential avenues of the action or drugs beyond those? And how far away from clinical trials do you think some of those are, if you can answer any of that?
So I'm going to kind of organize this into neural things and genetic things. Like, we know a lot about what the Fragile X protein does in brain cells, and we know about things it regulates. There are some targets to correct things that are misregulated because of the absent FMRP that have not yet been to people yet, because they were kind of new things and it took a while to develop. There are certain channel drugs that are moving forward. There's also a similar drug to the Tetra drug that works on phosphodiesterase. There are just a variety of things. There's some things that target proteins that work in protein synthesis that interact with FMRP. There are a variety of things that might be more potent than what we've been using thus far. We don't know. And so we'll need to test those things out. So those are all very hopeful. And, FMRP does so many things that it would be helpful for us to have everything to use all the things that work together. So it'll be good for us to work toward all these things. But the last couple of years have really brought a lot of new genetic strategies to Fragile X, or maybe we've been working on this all along, but we're just finally getting to the point where it's looking like it's not that long before these things will go forward. There are a number of gene therapies that are being worked on. There's a delivery of a mini protein that's being worked on. And the gene therapies include both a mini gene and the full gene. They're regulated in a variety of different ways. There's at least 4, maybe 5 or 6 different companies or groups that are working on this. There's the ASO therapy that Joel Richter and I discovered, this abnormal RNA. And there's a company working on suppressing that to increase FMRP. And so far, their experiments have replicated Joel's work and look very good. I think that's very promising. There's an RNA therapy out there that looks promising. So if you go from, DNA to RNA to protein, we have things in development at every one of those levels that are actually targeted to the key defect in Fragile X, which is low or missing FMRP. So all of these things are targeted at increasing FMRP in neurons. And so the fact that we have so many different options coming forward, and I don't know which one's going to hit the clinic first, but I think some of them might not be more than 2 or at most 3 years away. So I think it's very hopeful for those things in the future. But again, you can have the best drug in the world, but if you can't figure out how to demonstrate benefit correctly in patients with Fragile X, then you're not going to have a good trial. And that's where we need everybody to keep participating in things like the Forward March Project, which is designed to try to, develop, to try to understand the best kinds of outcome measures in Fragile X, and other, individual researchers' studies that are looking at outcome measures. So I think we're all in this together, and we need to keep working together and not get thrown by these negative trials and think, oh, we're never going to be there, because I can tell you that what's on the horizon looks a lot more powerful than what we've had before.
Let's hope one of them gets across the finish line soon.
Oh, yeah, because otherwise I'll be 100 and still be working.
So this next kind of ties right into that, and says that this is now another late stage trial in Fragile X that hasn't met its primary goal. And families are starting to describe the feeling as kind of like a trial fatigue, or like even like just, the constant letdown from repeated disappointments. Can you speak to the challenges of bringing a medication to market in Fragile X, and how having many different research options supported by FRAXA helps ensures progress continued despite individual trial failures?
Yeah, so I think we've had more failures than other specific diseases. Why? Because we've had more trials than other diseases, and because we started earlier than other diseases. We knew what our gene was. We had a lot of Fragile X research, a lot of it funded by FRAXA, where we found these targets in the brain and we went forward and tried to look at drugs. And we did that in a setting where no one had ever tried to develop a therapy targeted to the mechanism of disease in a neurodevelopmental disorder. So I always quote Einstein on this, who said, a person who has never, wait, maybe I have this, well, I'll come back to that. Who said a person who's never failed has never done anything new. So this was totally new. Our first round of trials that didn't go the way we wanted them to was, a result of doing something new. And we're learning, but, the Zynerba trial has been going on for a long time. And I don't think it has been the beneficiary of everything we learned, because it started way back before we had some of the more recent developments. So I know people get trial fatigue, but I think we're entering a whole new sort of generation of therapies coming up that are targeted to new and different and more promising things, especially the genetic therapies. And so we can take all the learnings we have from the trials that didn't work and apply those to the new trials. And I hope, I know that FRAXA has pushed forward a number of these things, and I think it's really just a matter of climbing every mountain, just like it was before when we had the previous failures. And the early things, some of it I attribute to just doing something new, and really, we've informed a whole bunch of other fields. The Rep people, the Angelman people have looked at Fragile X and said, okay, they figured out not to do that or to do that. And they've had the benefit of our, initial work and, the lack of success of some of that to build their clinical trials. And it's helping them move forward faster as well. So we were kind of first. So we're helping the world, the rest of the developmental disability field, figure out how to do this when there's a treatment available to them. So hopefully I'm being I don't, none of us like this news, but I don't think we want to wallow in it. We just want to say, look, okay, that didn't work. We're going to move on, and we're going to find the right strategy for the next thing.
And that's all the questions you have right now. We do still have a couple more minutes, so if you guys do have any more questions, feel free to use the Q&A at the bottom. We did have somebody to say thank you so much, Dr. Berry-Kravis. We appreciate what you do for us as the community, for the science, and for research. And we cannot thank you enough for this journey, being with us and leading us, and ultimately to a cure. It's kind of a nice little submission that somebody had provided for us.
Yeah, I wanted to add to that because FRAXA funded your first trial, Liz, way back. And boy, it's just been a long journey since then. And we have learned so much. And I agree with everything you said.
Yeah, that was 2002. And so we've had 23 years of learning.
Yep.
And hopefully we're going to get our degree pretty soon.
Oh, we will. We will.
So we'll see. But I want everybody, I hope everybody can stay optimistic. I know it's kind of devastating, like, to hear this again, but I think there truly are very hopeful things moving forward. And I'm hopeful for the Tetra trial. So, so just, keep pushing it.
Yep.
I mean, we're all in this together, really.
Yeah.
And I was muted. And that actually kind of ties right into the next, not a question that was submitted, but just kind of comment. It was actually from Anna from the National Fragile X Foundation, and they just wanted to extend their thanks to you, Dr. Kravis, as well as to FRAXA for putting this webinar together. They're grateful to be able to have a chance to understand RECONNECT trial news alongside with the Fragile X community. And let's see, we just had another question come in, and it said,
well, Anna was there for the Seaside fiasco. So she has experience, direct experience, personal experience. So hopefully one day this will not be the result. But I saw in the chat something about the Tetra trial going to kids younger than 9. Is that what you're talking about?
Yes, it was. Do you expect the tetracycline, I'm not sure, study to expand to children less than 9-year-olds or time for that?
Yep.
Okay, the Tetra study is not, tetracycline is an antibiotic. The Tetra study is of Zatolmilast, which is a PDE4 inhibitor. So I think if they have a positive result, it will definitely expand to the earlier group. They already have a development plan that includes that, but we need to see the adolescent study before we can know whether they're going to move. Their studies are closed right now. They're not enrolling any new patients. They're waiting to get results. And it always takes a long time to get these results completely the database is all clean and the results all analyzed. But I am positive that if they have a result in the adolescent study, they will, or the study that goes down to 9, they will be going to younger kids. Absolutely. That is their plan.
And girls as well.
And girls as well, right. But, the girls are just going to be in a separate study because it's so hard to assess them on the same scales as the guys.
I think that's it for tonight then. So I think I'll just take a moment and thank you, Dr. Kravis, for joining us, spending your time this evening, and just joining the community in general. And, even when on nights like this, when the news isn't the best. And to the families and community members, also thank you for showing up, for asking such thoughtful and heartfelt questions. And let's just continue to keep moving forward.
That's right.
Yep.
Forward.
And we hope for better news soon.
Yeah.
Thank you very much, Liz.
Okay.
Really
you're
appreciate it.
welcome.
Thank
Good night,
you, everybody. Good night.
Keep hope.